Next up, we're going to hear about some really wonderful advances in preclinical research for Sanfilippo. These programs are the building blocks upon which new treatments come about and allow us to move forward. These are discoveries that are foundational to the work that eventually reaches our children. Today, we will have a few speakers. First, we'll hear from Dr. Ryan Weiss, Assistant Professor at the Complex Carbohydrate Research Center at the University of Georgia, and along with his colleague, Dr. Nadja Zeltner from the Center for Molecular Medicine, also at the University of Georgia. They'll be sharing about their work to develop peripheral nervous system models for Sanfilippo. This is, really an understudied area, but so important. And as we heard, many of our children maybe can't tell us specifically about the challenges or the feelings that they have that may affect their peripheral nervous system, but it doesn't mean that they're not there. And that we heard from the Lindquist girls that, they do have different sensations in their body. And I think it's a really important area, so I'm excited to hear their progress. Next, we'll hear from Mr. Travis Moore and Dr. Patricia Dubot of St. Justine Research Center in Canada, and they'll be providing an update on their work to develop a small molecule for Sanfilippo diseases, focusing on Sanfilippo type C, but expanding to other Sanfilippo subtypes. And lastly, we'll welcome Professor Stephen Gray, Director of the University of Texas Southwestern Gene Therapy Program. And he'll be sharing updates on the development of an AAV9 gene replacement therapy for Sanfilippo type C. I'll hand it over to Dr. Weiss.
Yes,
I think that's good. Okay, thank you so much first for the invitation and giving us the opportunity to share a little bit of our relatively new work here. As Cara said, we are trying to develop peripheral neural stem cell models and then use those for, in the future, drug discovery for Sanfilippo syndrome. Next slide, please. Okay, so Cara said that already, but basically we are two labs here at the University of Georgia, and Dr. Weiss's lab is in the Complex Carbohydrate Research Center, and his work really focuses on complex carbohydrates as well as rare disease, and MPS is one of them. And my lab is literally connected to it, to the CCRC. It's in the Center for Molecular Medicine, and we're focusing on using human pluripotent stem cells to model diseases of the peripheral nervous system. And so we came together and sort of brainstormed a little bit how we can work on new model systems for MPS. Next slide, please. Okay, so let me introduce first the peripheral nervous system here a little bit. So the mammalian nervous system consists of two parts. The central nervous system, here indicated in pink, which contains the brain and the spinal cord, and then the peripheral nervous system, which are all other nerves in the body, which you can see here in blue. Next slide, please. So the peripheral nervous system is then again divided into two parts. The sensory nervous system, which is mainly important for sensing things like pain and temperature, stretch, but also sound and taste. And then the autonomic nervous system, which is all the nerves that you cannot control consciously, so they maintain the homeostasis of the body. And that's again divided into three parts: the enteric nervous system, which innervates your gastrointestinal tract, and then the sympathetic nervous system, which mediates the fight or flight reaction, and its opponent, the parasympathetic nervous system, which mediates the rest and digest. Next slide, please. So again, we were thinking of Sanfilippo syndrome, and are there PNS phenotypes in MPS III? Next slide. So we looked through the list of symptoms and also talked to Cara about that. And so some of them kind of stood out to us as being mediated potentially by the sensory or the sympathetic nervous system. So for example, crying, pain, especially in early childhood, crying might be an indication of pain. Loss of ambulation is an indication of proprioceptor problems. Dystonia as well could point to the sensory nervous system. Then also things like swallowing trouble, breathing issues, gut motility, hyperactivity, enlarged liver, and the coarsening of facial features may all be intertwined with sympathetic activation or not proper sympathetic activation. Next slide, please. Okay, so we are using human pluripotent stem cell technology to model these diseases. And so what are human pluripotent stem cells? I'm not sure what's the background of people in this room, virtual room, but however, so we can derive human pluripotent stem cells from two sources, either from the inner cell mass of the blastocyst, which you can see here on the left. I don't think they can see our, okay. And so this is basically a very early embryo that would have otherwise been discarded after in vitro fertilization. And so we can take these cells out, so the red cells in the green, inside of the green circle, and these are human embryonic stem cells. And these cells can be cultured in the dish at unlimited time, essentially, and that allows us to make as many cells as we want, which is great for screens and things like that. And so the second source is from human patient cells. For example, fibroblasts or blood cells, we can take these cells and we can transform them reprogram them, in vitro into what is called induced pluripotent stem cells. And for the purposes of our work, we basically make no distinction between iPS or ES cells. So they really behave almost exactly the same. So the second really important thing that these cells can do is they can be differentiated into every cell type of the body. And as you imagine, when the embryo develops, these cells will give rise to every single cell type in the body. And so with this in hand, we can now do cell replacement therapy. For example, you can think of Parkinson's disease as an example where patients lose specific cells in the brain. One can make these specific neurons and transplant them back into the patient, or at least as ongoing work. We can also use them for disease modeling, which I'll tell you in the next slide, and then do drug screening with these disease models. Next slide, please. So this technology gives us an opportunity to have untransformed cell types of patient origin, and it gives us the opportunity to make specific cell types, such as peripheral neurons, that are not possible to take from human patients for research. Next slide. Okay, so the human disease modeling with stem cells. So essentially what we do is we take a healthy person and a patient, in this case MPS, we take a biopsy, fibroblast or blood cells, we reprogram these cells into iPS cells and then differentiate them in vitro into a cell type that is affected in the patients. And here we were going to go first go for sensory neurons. And we then compare the two. So you compare cells, let's say sensory neurons from healthy people and from patients, and that will allow you to really dig deep into the mechanism of disease within this specific cell type. And then that can allow you to do drug screening. Next slide, please. Okay, so far we have 4 lines that we're working with. So we have 2 healthy lines, H9, which is a control embryonic stem cell line, C1.2, which is a healthy control iPS lines, and then 2 MPS lines, one from a 6-year-old female, which we were able to get commercially, and then one from a 3-year-old male, which we reprogrammed in our lab ourselves. And we're going to use both of these. Next slide, please. Okay, so the way we do this is basically we recapitulate embryonic development in the dish. So in this case, we're going from iPS cells to neurocrest cells, which are the progenitors of all the peripheral nervous system, and then to sensory neurons, which is the cell type that we started with. And we look at MPS phenotypes on each of this level. That's the next slide. Thank you. And 2 students in our lab, Tripti Saini, started this project, and Megan Shin has taken this over, and she's co-mentored in both of our labs. Okay, next slide. So we first look at phenotypes in the iPS stage. Next slide. So here you can see a typical picture of how undifferentiated iPS cells look like. On the top, you see healthy cells, and then in the bottom, you see the MPS cells. And you can see that there's really no difference at this stage. Next slide. So then we gave some of these cells to Dr. Weiss's lab, and they looked for several molecular phenotypes, such as SGSH enzyme activity, which is the enzyme that is mutated in MPS IIIA. And you can see that there's a drastic reduction in enzyme activity. They also looked at total heparan sulfates, which are massively increased, and this is what is stored in the lysosomes. And they also looked at a specific sugar, the N-sulfoglycosamine, which is also massively upregulated. There's much more in the lysosomes of the MPS IPS cells. So in general, we see at this stage no cellular phenotype yet, but we do see typical MPS molecular phenotypes. Next slide. So next we look at the neurocrest cells. So we take our cells and differentiate them into neurocrest. Next. Thank you. So you can see these typical in H9 on the top. You can see these typical neurocrest cells which come up as sort of higher clumps and dense ridges, these black dots that is indicated with the white arrow. And these neurocrest cells will stain for SOX10, which is a typical neurocrest marker. And also we can look at them by FACS for CD49D, which is another neurocrest marker. And so you can see when we compare this now to MPS that we see actually less neurocrest cells, both at the ridges and then less SOX10 and reduced numbers of CD49D. Next, please. We're also currently working again on the molecular assays. Here we're only showing you the enzyme activity, which is again or still massively reduced. Next. So we have a phenotype of reduced neurocrest cells, which again you could think of could lead to either reduced numbers, mostly reduced numbers of PNS cells. And then we have reduced enzyme activity and increased GAG activity, which we're not showing here. Next, please. So finally, we looked at the sensory neuron phenotype. So we finally differentiated these cells into sensory neurons. And so here you can see them on the left stained with BRN3A and TOJ1. And you can see again that we have less sensory neurons in the MPS lines. We then also started looking at LAMP1, which is a protein that's stored in the lysosomes. And it looks like that we get more LAMP1 in the MPS sensory neurons, and we're working on quantifying that. Furthermore, we seem to see increased swollen cell bodies in the MPS sensory neurons, which might be an indication on the cellular level of this storage, increased storage. Next slide, please. Again, here we confirmed at the sensory neuron stage the decreased enzyme activity, increased heparan sulfates, as well as the specific sugar. Next slide. So in summary, we see reduced sensory neurons, increased LAMP1, swollen soma, and reduced enzyme activity, as well as GAGs. And next slide. So finally, future directions for the cellular phenotype. So we're currently looking at these sensory neurons in more detail. We look at their electric activity. We will stimulate them with pharmacological agents that mimic, for example, pain. We will also and then see how they react. We will also do co-cultures, so we can see how target tissues that are innervated by sensory neurons will react differently in MPS. We then also look at sympathetic neuron phenotypes. So basically, we will do similar work that we showed with the sensory neurons, but now in sympathetic neurons. And specifically, we are interested in looking at co-cultures, for example, with smooth muscle cells that could be indicating of gut motility, and see how MPS innervated smooth muscle cells might activate the target tissue differently.
All right, next slide, please. So yeah, my name is Ryan Weiss, and my lab is primarily focused on studying heparan sulfate biosynthesis and as well as Sanfilippo. Next slide, please. So for this last part of the talk, I just wanted to show you some of the work that really coincides and works along nicely to developing these novel models for Sanfilippo syndrome. We're also interested in identifying novel small molecule agents that could potentially be used as substrate reduction therapies for not only MPS IIIA, but other MPS disorders that are caused by storage of lysosomal storage of heparan sulfate. So this was built on previous work. Next slide, please. Where genetically, this was work done by Jeff Esco's group at UCSD, where they actually crossed a mouse that was deficient in heparan sulfate. This is the mouse on the left. This is an EXT1 mutant mouse that is deficient with heparan sulfate with the sulfamidase knockout mouse, so this MPS IIIA mouse model. And the graph shown on the right, you can see there's a massive amount of storage. For instance, if you take the brains from these animals and look at heparan sulfate stored in these cells, the black bar shows a massive amount of storage in the Sanfilippo mouse. When we cross with these heparan sulfate biosynthesis deficient animals, you see this reduction in storage, and they actually showed a reduction in many of the hallmarks of the disease, for instance, inflammatory factor markers in the brain of these animals. So this led us to think about, what if we could find small molecule agents that could do this very same thing, for instance, targeting EXT1 expression to lower heparan sulfate assembly as a way to treat the MPS IIIA with a substrate reduction therapy. Next slide, please. So we developed a drug screening strategy where we could genetically tag, for instance, the EXT1 or EXT2 proteins using CRISPR-Cas9. So this is where we can knock in a small tag into the endogenous gene in the genome. In this case, we use this HiBit tag that was developed by Promega. So in and of itself, this tag doesn't really do anything, but when you mix it with the other portion of the enzyme, now it becomes a functional luciferase enzyme. So you can monitor EXT1 protein levels in real time by luminescence. And so on the right, it just shows that we have these cells that we developed that show luminescence, and they respond to changes in EXT1 levels, or if you just inhibit protein synthesis. Next slide, please. And so we did a drug screen in collaboration with Chris Rice, who is now at Purdue, but was here at UGA. And so we really initially wanted to take a drug repurposing approach and thinking, what if there are already FDA-approved drugs out there that we could take and repurpose them as substrate reduction therapy for Sanfilippo syndrome. And so this big graph on the right just shows our drug screening results. And you can see in the blue box, there's a ton of drugs. Each dot is a different drug that reduce EXT1 protein levels. Next slide, please. So we went through a lot of validation of all these hits that we got from the screen, and we ended up prioritizing these top 4 drug candidates. So we have a diverse set of drug targets: tyrosine kinase inhibitor, heat shock protein inhibitor, anti-tumor agent, and a YAP inhibitor. And I want to mention that all of these are clinically approved drugs that also get across the blood-brain barrier. So we're really thinking about, can we repurpose these drugs to treat the CNS phenotypes as well as the PNS phenotypes as well. Next slide, please. And so I'm just going to show a couple of validation experiments. And this is looking again at LAMP1 staining. So this lysosomal associated membrane protein, using an antibody against that. And we see when we age, for instance, fibroblasts from MPS IIIA patients, on the top left, there's a ton of LAMP1 that you can detect that's shown in green. And what you can show below it is just an example image, is that when we treat with one of our top hits, we see a drastic reduction in LAMP1 staining, suggesting that there's a reduction in this lysosomal storage phenotype. Next slide, please. And so finally, we also wanted to look at heparan sulfate storage, intracellular storage in these fibroblasts. So again, we can age them for 1 month. And on the top left graph, you can see in the red bar, there's a massive amount of storage in these patient fibroblasts. This was reduced with treatment of all 4 of these top drug hits that we found from our drug screen. And on the right, you can look at the non-reducing end biomarker, which is this N-sulfoglucosamine. This is all done by a liquid chromatography mass spectrometry technique that we developed here that builds off of work from Jeff Esco and others. And we see a drastic reduction also in the NRE as well in these. Next slide. And so we've started to expand this to other MPS III patient fibroblasts. So this is from 3B, 3C, and 3D subtypes. And you can see you see the characteristic storage, which is the tall bar in each of these graphs. And you can see that the drugs reduce the storage across all of these, which makes sense because they're all involving storage of heparan sulfate. Next slide, please. And so we're working with Nadja Zeltner's group to develop CNS models as well. So this is an example of prefrontal cortex neurons that Megan has developed. And we started treating our drugs with these, which is a more clinically relevant cell type. And we see a reduction in EXT1 levels, and we're still working on characterizing the effects of these drugs on these cells as well. Next slide. And so hopefully we're able to show you that we're developing new models to study the peripheral nervous system and MPS. And we're hoping to meld our drug discovery platform to find new ways to treat these underlying phenotypes, including lysosomal storage of heparan sulfate for these patients. Next slide. And so I want to especially thank the Cure Sanfilippo Foundation for funding this work and really allowing us to get all of this kickstarted. Also, other rare disease organizations, including CDG Care, have also helped with the drug discovery aspect, as well as NIH. And thank you all for listening.
Good morning, everyone. Thank you for joining us for our presentation. So I'm Travis, this is Patricia, and today we're going to talk about AVP6, a small, promising small molecule for treatment of Sanfilippo disease. So we're not only going to talk about cells, but we're also going to talk about the mice. Patricia's going to handle that portion. Next slide, please. So when you're looking at the therapies for MPS disorders or Sanfilippo disease, there's a number of challenges that we've heard about earlier and limitations. Number one is that of the blood-brain barrier, of course. So any drug that wants to help with any central nervous system problems needs to get past this blood-brain barrier. Additionally, mouse models don't always reflect the phenotype of patients. We have some really good mouse models out there, but of course, mice are not humans. They also have a lot of urinary retention that is not normally seen until later stages in patients. Additionally, if you want any sort of therapeutic to actually cause something to reduce the heparan sulfate or cause a restoration, you need to be able to stop the progressive pathology and able to inhibit or restore to a healthier level. Additionally, with the lack of established quantifiable biomarkers, we're not sure exactly where a patient is on the scale, so it's hard to quantify in terms of restoration. Next slide, please. So we do know, as I mentioned earlier, we do know a lot about the mouse models. Specifically, our lab has previously looked at a lot about the MPS IIIC mouse model. So we know a lot about the neurons themselves. So the number in the top left, you can see the number of spines in terms of wild type and MPS IIIC models. Over time, there's a decrease in the number of spines. Now, in terms of the excitatory neuronal network, seen with the protein markers VGLU1 and PSD95, individually, they are decreased on these neurons, as well as in the inhibitory circuit for VGAT and Geoffrin. We can see that there's a decrease in this mouse model, as well as just the number or the pool of synaptic pool present is significantly reduced. Next slide, please. So what this means, what these decreases in proteins means, is that there's actually a functional aspect to this. So overall, if we're looking at these miniatures, little m, excitatory or inhibitory amplitude or frequencies, this is a functional output of these decreased proteins. So you can see from the genetic aspects to these protein aspects showing these synaptic deficiencies to neural degradation and then further to behavior. Next slide, please. So we are looking at a potential therapeutic for the synaptic deficits, and this small molecule drug is called AVP6. And what's important about AVP6 is that it causes an increase of brain-derived neurotrophic factor. It's a really important molecule in the central nervous system, and specifically its mature form is important for neurogenesis, neuroplasticity, and long-term potentiation, all things that we hope to restore potentially in MPS lines. Now, we had previously done experiments in the MPS IIIC model with daily treatments of AVP6, and we saw really promising results for reduction or preventing the development of the behavioral deficits for hyperactivity, loss of fear, defects in memory and learning. Next slide, please. So, as I mentioned earlier, mice aren't always humans, so started to look at the induced pluripotent stem cell lines of patient-derived lines. So if you look on the left-hand side, anything I denote with CNT is a control line, and then you can reference from 3C-1 or -2. Each subtype of A, B, and C has two subtypes. So we had fibroblasts from patient lines, and they were converted into iPSCs. We then moved on to neural progenitor cells, looked at appropriate markers, and then further to test the actual drug, I either didn't add AVP6 or I did add AVP6, and we were able to look at these protein markers that we talked about earlier. Next slide, please. So first and foremost, if AVP6 is able to work on BDNF to increase this marker, then we should see that in our cell lines. So if you look at the top left corner there, they're the control lines. The first is non-treated, and there's BDNF red puncta along this MAP2 projection. That's just the neuronal projection. Now below that, you see when they're treated with AVP6, this is just control lines, there's an increase of BDNF. Now, if we go back to that top row, you can see as you go from controls to MPS IIIA to MPS IIIC and then down to MPS IIIC, you can see that there's a significant reduction of BDNF along these projections. But with AVP6, there's a restoration of BDNF on these projections back to healthy control lines. Next slide, please. Now, I talked earlier about those excitatory markers, VGLUT and PSD95, and the same markers were checked in these iPSC-derived patient neurons. So if you start on the top left again with VGLUT and PSD95, and you pan across for 3A, 3B, and 3C, again, you should be able to see that VGLUT is significantly reduced as well as PSD95. Now, with AVP6, we should be able to see specifically in the patient lines is that compared to their non-treated counterparts, they are significantly increased. And the quantification on the right-hand side shows that the VGLUT is restored across all these lines, and PSD95 is either restored or significantly increased compared to previously. Next slide, please. And then again, looked at the excitatory, and now we're looking at the inhibitory markers. And same thing again, we start at the control lines, and as you pan across for VGAT or Geoffrin, you can see the significant reduction in all the Sanfilippo lines that is restored on the bottom panels there compared to their untreated counterparts. And once again, with the quantification, you can see a very nice restoration of these markers in all the cell lines. So with that, this was looking at the full spectrum without D of the iPSC-derived lines. And now Patricia is going to talk about some initial experiments we started with MPS IIIA and MPS IIIB.
Hi everyone. So I'm gonna show you the first result we had about the AVP6 efficacy in MPS IIIA and MPS IIIB mouse model. So for that, Travis and I, we treated those mice for 6 months, from 1 month of age until 7. Next slide, please. So first of all, we analyzed the autistic feature because in MPS IIIA we observed this characteristic. So for that we use the sociabilization test. So for that we put the study mouse in the cage with 3 chambers inside. And first of all, we put the mouse for 5 minutes for habituation. And then for the session 1, we add on the right side a cage with a new mouse, and on the left side an empty cage. And in session 1, we measure the time of interaction between the studied mice and the empty cage or the new mouse to assess the sociability of the study mice. And then for the session 2, we add a new mouse in the previous empty cage. And in session 2, we assess also the sociability of the study mice and if the ability to mice to make new friends. Next slide, please. So here are the results. So on the top, you have the session 1 and the bottom session 2. So a normal mice prefer to spend more time with the stranger 1 compared to the empty cage. It's what we see here. And for the session 2, the normal mice prefer spending time with the new friend, the stranger 2. For MPS IIIA, we can see they don't have any preference for empty cage or the stranger 1 or the stranger 1 or stranger 2. So it means they behave with autistic feature. But for the treated MPS IIIA with AVP6, they behave like a normal mice. It means they are more social. For MPS IIIB on the right side, for the session 1, the results are not very similar because I did it automatically, so I need to redo it. But for the session 2, we have the same pattern than the MPS IIIA. So AVP6 prevent the autistic feature in a mouse model. Next slide, please. So then we analyze the memory and the learning process, because in MPS III mice there is a deficiency of that. So for that we use the contextual fear test, and with this test we can assess two things. First of all, the acquisition and expression of conditional or contextual fear. And then we can also assess the long-term memory process. So for that, we put the mouse in the new cage, new environment, new context. And the day after, we put back this mouse, the same mouse in the same cage, but the mouse received 3 mild electric foot shock. So every electric foot shock make the mouse scared. And we are able to measure the fear, because when a mouse is scared, they don't move. And we call that the freezing moment. And then to assess the long-term memory process, we put the mouse in the same cage at day 2 and day 7, but without any electric shock. Next slide, please. So here the results of, about the learning process. So for MPS IIIA it's blue, and MPS IIIB it's green, and the normal mice gray. So for the 2 mouse model we have the same kind of results. So for the normal mice, as we expected, after every electric foot shock, they are more scared, and so the freezing moments are higher. For MPS IIIA or MPS IIIB, we can see they are less scared. But for the treated MPS IIIA and MPS IIIB, they behave like the normal mice. So AVPC can prevent the deficiency of learning in these 2 mouse models. Next slide, please. And then we assess the long-term memory. So we compare day 2, day 7 to day 0. And for the normal mice in gray, you can see the freezing moment is high. So they remember that in that cage, they could receive a shock, so they're scared. But for the MPS IIIA or MPS IIIB, they don't remember. But for the treated mice, they remember, they associate this cage to the electric foot shock, and so the freezing moment is higher. So IVP6 can prevent the deficiency of long-term memory. Next slide, please. And finally, we test the short-term memory with the novel object recognition. So for that, we put the mouse in the cage with 2 similar objects for 10 minutes. And 1 hour later, we put back the mouse in the same cage, but we change 1 object. So at that moment, we have 1 familiar object and 1 novel object. And we measure the time spending with the novel object compared to the familiar object. Next slide, please. So here the results about MPS IIIA mice. I didn't have time to show you the MPS IIIB results. So for assessing the short-term memory, we calculate 2 indexes, the discrimination index and the recognition index, which are very similar. And the point above the dotted line means the mouse spend more time to explore the new object, so they recognize the novelty. So as you can see, for the normal mice in gray, the dots are above the threshold. But for the MPS IIIA in green, it's the average, it's on the threshold. So it means they don't have any preference with the novel object or the familiar object. They don't recognize the novelty. But for the treated mice with AVP6, they can recognize the novel object as a new. So it means that AVP6 prevent the deficiency of short-term memory in MPS IIIA mice. Next slide, please.
So that was a lot to go over between both cells and mice, but overall, we can see the effects of AVP6 treatment for the spectrum of MPS III, or at least a number of them. So in the cells, the cortical neurons that were derived from patient cells, we can see that the marker for BDNF, excitatory and inhibitory, are significantly reduced. These are all restored with the treatment of AVP6. And on the mouse side, because you can actually see behaviors from these treatments, we see that the sociability is completely recovered for 3A, or it's similar to increased in 3B. In terms of the contextual fear test, there's either a restoration or an increase back to the standard normal healthy mice, as well as in the novel object, they're able to now discriminate the difference between these two objects as compared to just the disease line without any treatments. Next slide, please. So thank you so much for listening. And I want to thank all of our current lab members and past lab members and funders, because no project is in isolation. Thank you so much for listening.
Thank you.
All right, it's a pleasure to be here with everybody today to present the work that we've been doing in collaboration with Phoenix Nest on doing a gene therapy approach for MPS IIIC. This was a treatment that has been in development by Phoenix Nest, and I agreed to kind of assist them with these efforts and do some preclinical studies in my lab. So, and I want to acknowledge Xin Chen and Thomas Dong within my lab, and also Alexei Shizhetsky, University of Montreal. So next slide. So this is a gene therapy approach. We're using adeno-associated virus as our gene delivery tool. And just for those of you that might not be familiar with this is a virus that doesn't cause any disease in humans, but it's been developed over the last half century as a carrier for healthy genes to use in gene therapy applications. There are hundreds of clinical trials now using different versions of AAV, and there are now several FDA-approved products that use AAV as the delivery tool for gene therapy. And so basically we're taking to engineer this as a carrier, remove all of the viral genes and replace them with some DNA, a gene cargo that we want to carry, and use the shell of the virus as a delivery vehicle. So if you go to next slide. If we, there's a wealth of information about using AAV, and particularly AAV9, to target the brain. And this is just an illustration of a study that we published in 2020 in mice, where we do basically a spinal tap or intrathecal injection of AAV9 carrying GFP reporter, and all the dark staining on these sections of the brain and the mouse spinal cord, all the dark spots are showing cells that are expressing the foreign gene that we're delivering. And it's still not targeting every cell. It's targeting a portion of cells. So when we're delivering the gene like this, if the gene product, the protein that's expressed, isn't shared between cells, then we need to think about this, especially as kind of a treatment rather than a cure with the current technology. So if you go to the next slide, that was results in mice. But when we look, trying to translate this to monkeys, it actually translates, when we're injecting relatively high doses of this, we found that it translates pretty effectively from mice to non-human primates. And this is just showing a study that we published in 2023, a small non-human primate study where we basically dosed the monkeys exactly how we would dose humans. And this was for a gene therapy approach for spastic paraplegia type 50 that's now in a clinical trial. And this was biodistribution data from monkeys where we surveyed 22 different regions of the brain and showed that the vector was distributing all throughout the brain, all throughout the spinal cord. And it was also going to peripheral organs. And that's something that we know about AAV9 is that, even if we inject it into the spinal fluid, a good bit of that vector gets out into circulation and delivers the gene to peripheral organs as well. So if you go to the next slide, specifically in the brain, this is just further illustration here where, and this was a non-human primate study that we did for CLN7 Batten disease, which is another disease that we've moved a gene therapy approach into a clinical trial. And basically just taking a section, sort of a slab in the middle of the monkey brain, focusing in on some areas that are kind of deeper into the brain structures, and all these little red dots that you're seeing are is the foreign gene being expressed in the brain of these monkeys, and all the little blue or purplish circles, those would label individual cells. So, again, we're not expressing the gene in all the cells throughout the brain, but we are distributing the gene, throughout the brain. So if we go to the next slide, this approach, and I just want to highlight this approach is being used now across a number of clinical trials, at least 2 or 3 dozen clinical trials right now for various diseases. Many of these are pediatric lysosomal storage diseases. And so there's a precedent here of applying this to, of using this approach for other diseases and then transferring this knowledge and this approach to MPS IIIC. So go to the next slide. And now specifically going into MPS IIIC, again, we were working with Alexei Shizhetsky, who developed this mouse model for MPS IIIC, that is this P304L variant that they've characterized and published. And so that was the mouse model that we used for this. So go to the next slide. The vector that was designed, that Phoenix Nest provided to us, used a design that's very similar to the design that we used for an AAV vector in a clinical trial for giant axonal neuropathy, as well as a clinical trial for CLN7 Batten disease. Uses a small minimal promoter to drive expression of the gene in a self-complementary AAV. So the idea here is to express a small amount of the protein, which should be enough to rescue the disease without expressing too much that it could lead to any kind of unintended toxicity. So go to the next slide. Yep, this is the design of our study in the mouse model of the disease. We also injected normal mice, and it's a bit complicated, but we injected mice either at 7 to 10 days old, kind of as a sort of best case early intervention kind of scenario, or we dosed mice at 4 months old when they would already have some disease burden. And then we collected a number of, sort of biological samples of blood, urine. We took down some mice early to look at expression and any histopathology. And then we maintained the rest of the mice longer term to either take down the normal mice at a year old to look at any kind of long-term safety issues, and then also taking the MPS IIIC mice out longer to look at efficacy. So go to the next slide. So in terms of body weight, we really didn't see, much of an effect on body weight across males and females out to a year. And this is, basically between normal and disease mice, the body weight wasn't really affected across a year. And so we didn't see any adverse effects with the gene therapy treatment either, as far as decreased body weight. So go to the next slide. Go to next slide, please. Okay. Yeah, in terms of survival, we did see a significant and kind of striking survival benefit in the mice, and this is really driven by phenotype in the MPS IIIC mice of extreme urinary retention, where, and I'll kind of go into this a little bit more, but they reach a point where they have to be humanely euthanized around 12 or 13 months old because of this issue, and we rescued that with treatment, and we'll go into that in a little bit more detail. So basically, we are seeing the survival benefit that's driven really around that particular issue. Importantly, in normal mice that received the high dose, we didn't see any adverse consequences in terms of survival. So all of this is speaking well to the safety of the approach. So go to the next slide. In particular around this issue of urinary retention, this is kind of an illustration here on the right, and this is when we're necropsying or kind of dissecting some mice. And this is a normal mouse on the left, and then a MPS IIIC mouse on the right. And you can see this like really swollen bladder that's filled with urine. And when we quantify this at 9 months old, we're finding that, and we can kind of get the mice to express the urine and then measure the volume here, where on average the MPS IIIC mice are holding on to about 0.5 milliliter of urine, and we're not seeing any urinary retention across any of our treated mice. So if we, and this is whether we treated mice early at 7 to 10 days old or late at 150 days old. So go to the next slide. And if we looked at this again at 12 months old, the urinary retention phenotype in the MPS IIIC mice that got the vehicle injection got worse. Now they're retaining, up to a milliliter of urine, which is a huge amount for a mouse. And then if we treated the mice early with either the low or the high dose, we're completely rescuing this phenotype. If we treat the mice later at 4 months old, then we start to see, a little bit of an incomplete rescue where, we are improving the phenotype, but now we're seeing this dose responsive effect where the high dose is working better than the low dose. So go to the next slide. We wanted to look at behaviors of the mice and really cognitive behaviors that we might rescue. And to be honest, the mouse model, we had trouble quantifying a lot of behavioral deficits, but there was one test that was quite robust for us where we were able to see a clear behavioral phenotype. And this is called a novel object recognition test. And this is basically where, mice are inherently curious, so you can put in a novel object into their home cage, and the mice will want to explore and investigate that new object. It's just a measure of their curiosity. And that does relate to cognitive function. So if we go to the next slide, when we quantified this, and the way that this is expressed here is we have this discrimination index on the y-axis, where a 0 would mean that they spend just as much time interacting with the novel object as they do just wandering around the cage. And so if we go above that dashed line, above the 0, then that means that the mice are interested in that object. They're exploring that object, which would be typical mouse behavior. So at 6 months old, we start to see that emerge where the MPS IIIC mice injected with vehicle are kind of scoring lower on this discrimination index than the wild type or the normal mice. And then it gets worse when we look at 9 months old, where there's really this deficit that is emerging, where the MPS III mice injected with vehicle are doing poorly on this task. But we can look at all, both of our treatment groups, the low and the high dose, whether mice were dosed earlier in their disease or later in their disease. We see this nice rescue of that effect where they're behaving like normal mice. So if you go to the next slide, the last thing that we looked at, and again, this is in collaboration with Alexi's lab, where we took a small number of the mice down and necropsied them at 8 months old, and we sent tissue, brain, spinal cord, and liver samples to his lab, and he measured the enzyme activity, where we're seeing this partial rescue across all 3 tissues, where we're increasing the amount of HS or HGS NAT enzyme activity, not back up to wild type levels, but we think, and I think our behavior and survival data is indicating that this amount of rescue is actually enough to drive this phenotypic rescue. So if you go to the next slide, really quick, our summary is that we're seeing improved survival of the MPS IIIC mice. The urinary retention is apparent in these mice, and we're rescuing that phenotype in a dose-responsive manner. We're also rescuing this novel object recognition phenotype, pointing to a cognitive improvement or normalization of the mice. And then also we're showing that the reduced enzyme activity in these mice is being partially rescued with treatment. So if we go to the next slide, I just want to acknowledge my team. And again, this is mostly Xin Chen, who's in the blue shirt on the right here, as well as Thomas Dong, who's not in this picture. But everybody in my lab kind of pitches in any way that they can help. And then we can jump to the next slide, or I'll just acknowledge, and I'm acknowledging a lot of people because it's really a lot of work went into building this platform approach of using AAV9 injected into the spinal fluid as kind of a translational gene therapy approach to target the brain. But this project is exclusively funded by Phoenix Nest, and I'm very grateful for them. And I can end there. Thank you.
Thank you. Thank you to all our speakers. Maybe all of our heads are spinning at this point. That was some amazing, amazing work you guys are doing, and a lot of incredible detail. Where to begin? Let's start with the AVP6 team and Dr. Dubot and Mr. Moore. You noted that one of the action that you think for this molecule is that it increases brain-derived neurotrophic factor. Yeah. And I was wondering, do you think there are additional mechanisms of action at play here, or is that really the one driving the beneficial effects you're seeing?
It's a really good question.
Yes, I'm working on the mechanism of this drug, and for now, I can tell you that AVP6 acts on the melanocortin receptors, 3 or 4, and the increase the phosphorylation of the CREB, which is the transcription factor of BDNF. And also, this molecule can reduce the inflammation as well, in decreasing the phosphorylation, of NF-kappa B.
Yeah.
Okay.
And we've seen some really good results in the mouse models for decreasing that sort of inflammatory response as well. So,
understand. Okay, yeah. I was curious as to what you think might be the clinical translation of biomarkers to a product like this, as opposed to something that's targeting heparan sulfate. We are probably looking at different biomarkers for this type of drug.
Yeah, because our molecule doesn't affect the heparan sulfate level. So
co-treatment type idea would be the best.
Yes,
in the future.
But maybe with the BDNF, I don't know if we can measure it after treatment with the AVP6 in patient, maybe in CSF. Could be a good marker. I don't know.
Thank you. Thank you. Okay, University of Georgia team. I
Yeah.
wonder if you are seeing consistent EXT1 suppression of heparan sulfate across the Sanfilippo subtypes, kind of consistently for each drug, or are the different subtypes responding to the different drugs differently?
Yeah, that's a good question. Yes, we've looked at mostly fibroblast models right now, and they seem to all be responding similarly to these, suggesting that the targets of these drugs, which we're still figuring out the underlying mechanism, but we do see changes in autophagy. So some of these drugs seem to stimulate autophagy, which we know in some of these MPS III diseases there's a suppression of this, and so. Yeah, we're seeing similar effects across the different 3 A, B, C, and D models that we've looked at so far. And we don't have an EXT1 antibody, but EXT1 mRNA levels are down. And then, like I said, we're looking at other markers like autophagy. These also seem to affect TFAB localization. So I think they seem to be stimulating lysosome biogenesis as well. So it's possible that multiple, maybe potentially positive impacts of these drugs might be leading to downstream changes in phenotype. Obviously, what we want to do is put this into an animal model. And what's cool is that these drugs are literally already in human patients. Some of them are being, for instance, investigated for dementia and other things. So CNS-related diseases. So that's something that we're wanting to work on in the very near future.
Great. Yeah, a lot of opportunity for, like you said, potential combination approaches as well with, the primary enzyme targeting approach that Professor Gray spoke about. And I wanted to ask you, Dr. Gray, given that type C is a membrane-bound enzyme, I wondered how that affected the approach that you decided to take and using, quote unquote, a milder promoter. Did that have to do with the fact that it isn't able to be secreted?
Yeah, I think all these things factor in. And I should say, we've worked on diseases that have secreted enzymes like CLN1 Batten disease or Krabbe disease or AGU. And in those cases, we try to use a strong promoter because we're trying to make each cell be a factory to make as much of the enzyme and share it around as we can. In the past, when we've worked on CLN7 Batten disease, which is, about 3 years into a clinical trial, that's a similar situation as here, where it is a membrane-bound enzyme. And in that case, we're just trying to express enough protein to rescue the cell that's targeted without trying to really just blow out the expression, which can lead to some toxicities. The other issue is we're trying to use the self-complementary design of the AAV, which makes it much more efficient. And in order to do that, we had to use this smaller promoter. So everything really tied together there.
Okay, I understand. Do you feel like from the results that you have so far that you've identified a clinical dose to move forward with? Yeah.
And it's really aligns with the dose that we're using in other clinical trials with this vector and this approach. So it's really what we expected to see.
Okay. So would you be going with that high dose or the low dose? Which
the high dose, yeah.
The high dose, okay.
Yeah.
Right.
Yeah, and that was one of the things that came out from our studies where in a lot of measures we didn't necessarily see a dose response, but I think that there were indications that the higher dose, in some of the measures, the higher dose was more effective. So it kind of justifies using that higher dose, which conceptually would make sense here.
And then typically the patients would more align with this later treatment paradigm that you saw in the mice versus the earlier treatment. So you want to cover for that.
Yeah, and I think that the earlier treatment age is really probably modeling more like treating infants that are even pre-symptomatic. Yeah.
Okay. Wonderful. Well, thank you all so very much. Really appreciate the work you're doing and can't wait to hear more updates.
Thanks for having us.
Thank you.
Thank you.
Thanks,
Yeah.
everyone.